Hi everyone, welcome to neuroscience webinar. My name is Rafael Novak and I will be your host today. Today we'll be talking about brain oscillatory activity associated with space flights, and I'm very happy to welcome our guest speaker. We have today with us Professor Fernando Maestú from Complutense University of Madrid. Hi, Fernando.
Hello.
Hello, Fernando. So let me quickly introduce our speaker. Let's say quickly, because he got huge curriculum. Fernando is a full professor and director of the Center for Cognitive and Computational Neuroscience at the Complutense University of Madrid. He obtained a degree in psychobiology and a Ph.D. in neuroscience from the Complutense University of Madrid. And his doctoral research focused on investigating episodic memory disorders in patients with mesial temporal sclerosis before and after epilepsy surgery, and utilizing EEG and MRI for this. Following his PhD, he pursued a postdoctoral fellowship in MEG, in magnetoencephalography, with Professor Andreu Papanicolaou at the University of Houston. And then, upon returning to Madrid, he initiated the establishment of MEG laboratory, and it was year 2000, and actually marking the first of its kind MEG lab in Spain. Subsequently, he secured a permanent position at the Complutense University of Madrid, and in 2009, Fernando secured funding from the European Union Commission to acquire a new MX system, and this financial support enabled the establishment of the state-of-the-art MEG lab at the Center of Biomedical Technology in Madrid. Finally, in 2017, he achieved the status of full professor at University Complutense of Madrid, and currently, so he serves as the director of the Center of Cognitive and Computational Neuroscience at UCM, and also holding the position as an invited professor at the University of Houston in Texas. And over the past 2 decades, Fernando's research has primarily revolved around the utilization of EEG and MEG techniques, mainly with the realm of Alzheimer's disease, but more recently, he started a line of research regarding the relationship between the brain and spaceflights. And this is actually what we are going to talk about today. So thank you very much, Fernando. The stage is yours.
So thank you so much for inviting me. It's a great pleasure to be here. I have been attending many seminars from neuroelectrics, so it's I'm very happy to be part of the club of speakers, previous speakers. So the idea of this seminar is talking about this brain oscillatory activity associated with space flights. There's I have divided the talk in 3 main topics. First, talking a little bit about the risk factors associated with space flights, and then I will talk about previous brain morphological and functional studies indicating differences in pre- and post-flight in astronaut and cosmonauts. And finally, I will talk, of course, about the EEG in the space and how the brain oscillatory activity changes as a function of these space flights. So, talking about the risk factors, of course, there are many risk factors because we human beings are used to live on Earth, and our nature is from here. But of course, as we go outside, we are in a different environment, and we can get we have some risk factors for our brain, which is, for example, radiation. The cosmic and solar radiation can affect our brain, but as well things like microgravity. It could change us, as we will see. Can change our morphology, even the CSF distribution, and that can cause some problems in the astronauts and cosmonauts. And of course, the idea of being there alone. I mean, you are with 5 people, 4 people typically, but you are mainly isolated from your normal social activities and your family and your relatives. So it is a very special situation, and that can affect as well mood, behavior, and actually the brain. It is true that all these can cause, of course, changes in your ability to perform a cognitive task, and this is very important during the space flights. Nobody knows whether radiation, weightlessness, or isolation, or sensory deprivation, what of these factors is influencing more the potential cognitive decline or cognitive changes that can happen during the space flights. But of course, all these factors can contribute. But there is one syndrome that is appearing relatively often in astronauts, which is the space-associated neuroocular syndrome that affects the eye, and that seems to persist a little bit even after the landing. So it is interesting, this cognitive idea, because astronauts, there is a very interesting paragraph in the paper of Stan and Kuhn saying that 2 astronauts in the Apollo 14, that Mitchell and Seyfarth, that they had to go to the Cone crater to get some rocks and material there, but they were not able to find it because they were not able to follow the map. They were not able to find the place, and they were actually really close to it. They were just 30 meters away from the crater, but they were lost. So it seems like probably the visuospatial attention and memory can be affected by these conditions, probably stress or radiation or whatever. So it's affecting not just the brain, but it's affecting probably the missions that they have to take during the space flights. So there are many countermeasures that can be done to improve cognitive functions and to try to avoid these potential effects of the space flights. But they are being investigated, and some of them are very successful, I would say. The animal models have been providing very interesting results in relation with radiation. So there are some studies that are trying to simulate the level of radiation that astronauts can get in the ISS, and see what happened in the brain of these animals. And they showed a really interesting finding, which is an increase of amyloid, specifically in male mice with a high dose of radiation. You can see here, this is the hippocampus. You can see here how the number of amyloid plaque is increased in conditions with high dose of radiation. And this happened mainly in male and not in female. This is interesting because this is a finding that is repeated many times in many other different animal models, which means that probably female are more protected to radiation than males for some reason. So this is the first study, but this second study was finding very similar results. You see here different levels of radiation, and again in the male animals you see an increased number of amyloid plaques as a function of radiation. There is an increase clearly in all areas of this A beta 40 and 42 in the hippocampus and other regions of the medial temporal lobe. So meaning that probably radiation is causing some sort of alterations that produce this accumulation of the amyloid. And of course, there is other physiological activities or other cells that are affected as well, which are the microglia. And seems like the high doses of radiation induce a microglial activation. Actually, in this study, they mixed the study of the microglia with some other behavioral data from the animals, and they, as a function of radiation, they diminish social interaction, increase anxiety-like behavior, and impaired recognition memory. So it seems like this the radiation, this as an isolated factor, can cause many different factors in the brain: activation of microglia, increase of amyloid plaques, and of course some behavioral changes that are very important for having this in mind for future as a factor we have to be protected from in the space flights. So, and finally, this is a very interesting study as well, in which they indicate that the high doses of radiation reduce dendritic complexity. You can see here no radiation, and here in the down part of the slide, high doses of radiation, and there is clearly a diminution of the complexity of the dendrons. So it is clear that radiation is making some sort of changes in the brain of the animals that are in these conditions, and probably they are something similar can happen as well in the human brain. So let's move then to the studies that has been done with MRI. That there are some studies that have been done before and after the space flight. So the astronaut or the cosmonauts are having MRIs, DTI or functional MRI or volumetric MRI or T1 to see how space flight affects functional or just the morphological aspects of the brain. So going to the volume, there was a very famous paper published in the New England Journal of Medicine in 2017, not very long time ago, but it was interesting that they showed that the central fissure practically disappeared after flight. You see here, this is the MRI from one of the astronauts. You see here the central fissure, but this central fissure practically disappeared after the space flight. Probably this is because of the shift of the changes in the cerebrospinal fluid due to microgravity. But this is another factor then to take into account. It's not just radiation that changed the brain, is as well the microgravity. There is a subsequent study that is really interesting in my view, published by Gillins et al. Is indicating that the microgravity induced the upward shifts inside the skull. So meaning that the cerebrospinal fluid decreased in the top part of the brain and goes down to the down part of the brain. That is indicating that actually the brain goes up, and all the fluid we have in the top part goes immediately down, and these changes in the cerebrospinal fluid can cause big problems, as you will see. The good news is after 7 months after landing, there seems to be a gradual recovery of the initial situation. So the CSF goes to normal situation as it was before the space flight. But during the space flight, you have these changes that some of them keeps, because there are some studies, follow-up studies, that are indicating that it's not fully recovered, but it goes more or less to the similar situation than in the pre-flight. But it is important as well that in this very good study, they showed gray matter changes and white matter changes. So in the gray matter changes, they showed that there is an increase of gray matter in the top part of the brain, but this is not because there are more neurons during the space flight. It's because the brain goes up to the top part of the skull, and that produces a kind of crowding there of material, of gray matter, and that induce more volume there there is a reduction actually in the down part and increase it in the upper part. But to me, the most interesting changes are the ones related with the white matter. It is very interesting that in the cerebellum and in the motor areas, as well in some basal ganglia areas, there is a clear plasticity, probably due to the loss of weight. I mean, having this microgravity context, you probably need to resituate. You need to recalculate your weight and your motor activities, and that induce changes in the cerebellum and in the motor areas and the basal ganglia as well. This is clearly seen in this picture, the areas where there are significant post-flight changes in the white matter. So increased white matter. So this is that was radiation, microgravity, but what about isolation? So this interesting study that indicates that after 520 days of isolation, it's a long period of time for being isolated in an analog. So this study is done on Earth. There are many places where you can do studies where you simulate the conditions of the ISS or simulate the conditions during the space flights. They are very good places for doing that. So in an analog, you can stay for more than 500 days, isolated. And what they do is an MRI before and after, and they saw it clearly a reduction of the anterior cingulate cortex and superior fasciculus tracts values, FA values, due to the isolation. So probably the social interaction is needed in our brain, and when we get rid of that social interaction, there is changes that is being produced in the brain connectivity, in the structural connectivity as well. So we have radiation, we have microgravity, and we have isolation that change the brain or are inducing important changes in the brain. In this studies is done with cosmonauts, so there is not an analog. This is real data from cosmonauts before and after the space flight, and you can see that this cerebellum plasticity is repeated in this study. You see clearly how the tracts in the cerebellum are increased after the 6 months at the ISS. So the brain needs to change probably the motor condition, the precision of the motor activity, and that's why the cerebellum change as in this independent study. But there is as well a decrease in the tracts. As you can see here, in many other regions. And the most interesting finding from this study is the idea that after a long period of time, like 7 months after landing on Earth, these changes in this decrease of tracts in the structural connectivity are still there after months after landing. So meaning that probably the recovery of this white matter tract changes is slower than other functional changes. And this is interesting as well, because probably we will need interventions that recovery much faster the situation of these astronauts and cosmonauts. Regarding the functional connectivity done with fMRI, you see here as well in this in cosmonauts that there is a reduction of the connectivity of the posterior cingulate cortex in comparison with other regions of the brain. This is a seed analysis where you put a seed in a particular region of the brain and then calculate how many connections you get to other regions of the brain, and they did it before and after. And clearly, in the post-flight condition, there was a reduction on the number of connections in this particular region of the brain. And as you know, probably this is a very important region for connecting with the medial temporal lobe and with the other prefrontal areas. And you see here clearly, these are individual trajectories. You see each of the lines is each of the cosmonauts, and you see the majority of them. So with a decrease in connectivity in the post-flight conditions, while the controls, there's another group that didn't go to the ISS, they stayed stable, more or less stable, or there were no differences between before and after this time period. Okay, so in the same study, they did a follow-up to see whether this functional connectivity decrease is just right after landing. So, but they did another MRI 7 months after landing, and they showed that this decrease in connectivity was still there even 7 months. And this happened as well. I didn't comment, but this happened as well in the thalamus. There is a reduction in the post-flight, and it continues 7 months after landing. So it seems like there is no much recovery in some structural and some functional connections that needs probably help to these astronauts to get better and to get better faster if there is no natural recovery after the space flights. So let's then move to the EEG, because MRI is providing fantastic results. I really like these results from MRI, but the only technology that really can evaluate brain activity during the space flight is EEG. So it's the only one that we have to evaluate the brain. We have been evaluating cardiac activity, many other factors in the body, but brain was not really evaluated during the space flights or not seriously evaluated. And that's why we think EEG can make a big role during the space flights. So let me give you a very brief story of the EEG in space, really fast. And I have to thank Professor Michael Funke, who is the person who knows better the story of EEG in the space flights. I think he's the one in the world that know more about the history, and he did this very interesting slide, and I will just comment on it. He's writing a paper on this, so he will be published soon. So he in the Vostok 3 and 6, there were already EEG, and that was in the 60s. They were evaluating EEG already. They didn't publish anything until very late. In the Gemini, they have as well EEG, in the Soyuz. So in the Mir, you have many recordings of EEG, but they never really published it like scientific papers. They published reports or with qualitative data more than quantitative. But EEG was there just to monitor brain activity, and nothing serious happened. But they were not really paying attention or scientific attention to the data. And it was when the ISS comes that there were a series of studies that were doing EEG with more electrodes, because the previous ones were just 2-3 electrodes in some part of the brain. And then right after that, for example, the Neurospat study, NeuroCog, they were using more electrodes, more than 15, more than 20, and some of them even 64 electrodes. Nowadays, there are studies being done with high density EEGs with different technologies. So this is the first study, and Professor Funke was able to get this picture from a book published in Russia. you see clearly but you see the EEG data here in combination with EKG and pneumogram and electroculography. So you can see that data is there, and they were using that probably just to monitor that nothing serious happened to the brain of these astronauts. And you can see here in the same museum, you see they show it physiological parameters, and EEG was collected at least in four of the cosmonauts. But there is a very interesting qualitative report in this study. This was just an internal qualitative report, but they say that alpha right decreases. They saw that the voltage seems to decrease. It was completely qualitative. It was not a quantitative study, but they say, okay, it seems like alpha decreased. And this is interesting to us because they found this in 1965, and we are finding very similar results right now with the EEG. But it's a pity that this data is not available anymore, because it will be very interesting to compare those EEG recordings and the ones we are doing now. In the Gemini 7, I wanted to comment on this because this is the only mission that had EEG during ascent. So when they were from Earth to the outside, they were recording EEG all the time, and this is the only experiments where we're doing that. Unfortunately, this data is not available anymore, but it's the only recording we have during the whole process from Earth to the moon. So it's interesting. It will be very interesting to repeat this and to see or reanalyze this. We are thinking on doing some analysis, trying to digitize this data and try to analyze it, but let's see how it works. And of course, in the Skylab, there were EEG. They were using then more like cups, like the ones we are using in the modern EEG. This is interesting that the space flights are used always for testing new technologies, and it's true again. The EEG cups were tested at that time in these astronauts, and you see here again the EEG recordings from these astronauts, and it looks really well. I mean, they are really clean data, so it doesn't seem like it is a very noisy environment in the up there. And of course, the mirror where you see recording less electrodes, but still having this cap with 6 electrodes. It was interesting as well that they were recording this data. And in 1998, the Neurolab with the digital EEG, you see here this oscillatory activity recorded as well in this age. So modern EEG technology comes, and in the 2000 decade, you get more high density EEGs with better data and better experiments. The one done by Professor Cheron is fantastic data and fantastic experiments. He has published several papers on this study, and it's providing really interesting results. For example, the loss of the theta synchronization because of the weightless. And this is a very interesting study done by Professor Narici as well. They put a radiation particle detector around the head of the subject to evaluate whether these particles are affecting or not the brain. And at the same time, he was recording EEG to try to understand how the dynamics in EEG can be affected by radiation in particular subjects. So they have this very fancy device here trying to evaluate these particles, as well as they were evaluating EEG at the same time. This is nowadays EEG recordings, as you can see, from different manufacturers that are providing experiments in the ISS and in analogs as well. There are some dry electrodes that are being tested as well as potential EEG devices. Particularly, I think we need more high spatial density electrodes than for evaluating the whole brain. At least in the beginning, we need that. And the Chinese are using a very high density electrode cap as well for evaluating brain activity in their own space station. And this is the last one regarding this historical part. This the Axiom 1. This is a commercial mission have been using this EEG, a high density EEG device. So nowadays, a majority of the EEG recordings in the ISS or in orbital missions are being used at high density EEGs, which are providing better data and probably better results for everybody. So one question that is very important is because when we were writing grants for doing this research, many times we got an answer from reviewers saying, okay, it should be impossible to record EEG in the ISS because it's a very noisy electromagnetic environment. So you can't record electrical activity properly in that environment. So what we had no answer at that time to the reviewers. So we decided to do data analysis, which is always answering all these questions. And as you can see here, you see in blue the post-flight, in green the pre-flight, and in orange the in-flight EEG recording. This is a power spectrum, and you clearly see the 50 hertz electrical noise in the pre and the post-flight, but there is nothing in the in-flight recording, because the electrical activity you have there is DC. In the ISS, you have no electrical contamination then to this frequency analysis. And you see that the signal is much better probably in during the in-flight recording than during the pre and the post-flight recording. In both eyes open and eyes closed, you clearly can see the differences during the in-flight recording. So this paper done by Patrick Fiedler, which is a brilliant mind person, did this data quality analysis to demonstrate that data quality is really high, really good in the ISS. This is a very interesting finding from Cheron studies, and he did a comparison. What is the difference between eyes closed and eyes open before and after the during Earth and during the in-flight recordings. And he calculated the differences in alpha power in eyes closed and eyes open. This difference, and he found more or less very typical results in the 10 hertz. In the pre and in the post flight, but during in-flight there is much bigger differences between eyes open and eyes closed. This is an interesting finding that I will comment on it after. So, and this is the one of the papers he published, that the phase-locking theta alpha oscillations was suppressed in weightlessness conditions. And so in the ISS, the microgravity induces this loss of the phase-locking theta and alpha oscillation in the virtual 3D tunnel condition that is a task that he did with the astronauts during the space flight. This is as well very interesting, because the proprioceptive sensations for being without weight is affecting as well our brain activity and our ability to understand the conditions. So, but in the Cheron design, and we asked him for his data to try to calculate with other techniques, functional connectivity, power analysis during resting state, to see if we can more or less describe what are the differences between the pre-flight, in-flight, and post-flight. Cheron did a fantastic design of this study. He recorded the electrical activity 3 times before the going to the ISS, 2 times during the ISS, 8 days and 54 days after they were in the ISS, in the International Space Station, and then 3 days, 7 days, 16 days, and 20 days after they landed again. So we were able to analyze these differences. I have to say that the preliminary analysis didn't provide differences between the 3 pre-flight and the 3 post-flight conditions. So we averaged this data together because there were no difference in the 3 times the pre-flight and the post-flight. There was no changes, indicating again that there is, if something happened, there is no recovery after 20 days after landing, as it happens as well with the structural MRI and the DTI as well. So it is important to say that this, because some reviewers as well say, okay, EEG is not reliable. I mean, you can do one measure one day, and then you do another recording 2 days after, and the brain changes completely. So it's not reliable. You cannot do longitudinal studies with EEG. And that was an important critic claim to the EEG. But we did a study indicating that the electromagnetic activity is stable across time. So we evaluate 16 subjects 3 times the same hour of the day, and we found that there is, especially for functional connectivity metrics, we saw an intra-class correlation across sessions, very stable results in different frequency bands, indicating that clearly that the EEG data is reliable, and you can use it as a dependent variable for longitudinal studies. So this is important to say. But what we have found during in this comparison between pre-flight, in-flight, and post-flight conditions. These are the 5 astronauts. Unfortunately, always these studies are reporting very few number of subjects. You can't have like 100 of them, because just 10 to 12 subjects goes to the ISS every year, even less than that. So it could take a whole career of a researcher to get a good sample to publish. But still, with these 5 astronauts, we found subject by subject replications or subject by subject profiles, which is always very, very good in EEG as well. So you see here in the pre-flight, they were showing more or less the same values of the alpha power in the pre-flight condition. But during the in-flight condition, there is a clear reduction in all of them. In all of the 5 subjects, you see a clear reduction of the alpha band. And more important, this alpha band power did not recover to the previous situation after landing, after many days after landing. So there is clear statistical differences between pre-flight, in-flight, and pre-flight, post-flight, and in-flight and post-flight, meaning that there is some sort of recovery, some sort of natural recovery, but probably this needs more effort. This needs something that helps more to get recover this alpha rhino, which is very important, as we all know, for cognitive functions. And this, of course, these effects were found in the default mode network, in the regions of the default mode networks, mainly. In other regions, this profile was not found, but it was clearly found, especially in the default mode network. So in eyes open, we did eyes open analysis as well. We found similar results. You know how hard it is to, to really describe the alpha band in eyes open, but it still is there, the effect of reduction of alpha that is still reduced in the post-flight conditions. So what about the functional connectivity? That is another important aspect. Alpha power is being reduced during the in-flight, but what about phase locking values of the phase of the signal, the synchronicity in phase, how it is behaving during the space flight? So there is a differences between the pre-flight and the flight condition. You see here clearly a reduction of the functional connectivity in the alpha band evaluated with Ph.D. During the flight condition. There is some sort of recovery, but not as exactly to the same values that we found in the pre-flight. There is better recovery in the functional connectivity than in the power, exactly in the same regions of the default mode network, but it's not fully recovered in the eyes closed. In the eyes open, we found differences, but you see they are not as spectacular as in the eyes closed. So there was we don't know why it happens. So there is an alpha band reduction. We know alpha band reduction typically is associated with in many studies with cognitive difficulties like attentional, memory, spatial memory, inhibition. So there, alpha band is a good biomarker for some cognitive functions. So yeah, if it is reduced, probably it's something we have to deal with and we have to evaluate better. So we were very lucky to contact with these guys that published this fantastic paper, Brown et al. And they were doing a bed rest. They are evaluating EEG in an analog on Earth, but they are simulating more or less microgravity. So this situation where the brain CSF can shift and can change the position and probably affects the brain. And what they see is when they were doing EEG is a reduction of the alpha band and theta and delta, so many other frequency bands during the bed rest condition. So when they are in this bed rest, they have reduced the alpha band. It is interesting that right after, when they recover the position, the alpha band recovers the power immediately, and the alpha and the theta and all frequency bands recover the power immediately. So seems like microgravity is affecting the power of the frequency bands, but it is some sort of recovery right after when they recover the position. So what we did is to ask the data to these researchers to compare, to try to compare our data during the in-flight recordings with the bed rest to see what are the differences in this reduction of the alpha band. You see here, this is the bed rest data set, and you see the before, during the bed rest, and after the bed rest there is a decrease in alpha and then increase. But in the astronauts data set, the reduction of the alpha was higher, but the alpha power was not fully recovered when they landed. This is an unpublished data, so we still have to write the paper and do more analysis. But it seems it is a different. The bed rest and the actual in-flight conditions are a bit different. This is a direct comparison. You can see here the alpha power during the bed rest and the alpha power during the in-flight EEG recordings. And you clearly see that is the reduction of the alpha is much higher during the in-flight conditions than during the bed rest. So bed rest is really good in simulating the microgravity, but probably in the ISS, there are other factors that are affecting the alpha power, not just microgravity. Microgravity will be one of them, but probably radiation, isolation, or some others are affecting the alpha power as well. And functional connectivity is more or less the same. This is the bed rest condition, and then the ISS alpha functional connectivity, and you see there is much greater reduction of the functional connectivity in the in-flight, the ISS condition. So, of course, EEG can be used not just to give very bad news to the astronauts, can be used for many other things like BCI. There are many other ideas that can be really interesting for EEG in the spaceflight missions. But one of them could be to understand who is on fatigue, because this is something that astronauts and cosmonauts are reporting many times. They are saying, okay, we don't really notice if we are on fatigue sometimes, because we are so excited doing our activities, our missions, and sometimes we don't notice this, but the EEG can notice, and this is interesting to monitor the brain activity during the whole time, because we know that there are parameters, as you can see here in alpha reduction, could be due of to fatigue. This could be associated as well to fatigue. So this is interesting to use EEG not just to evaluate consequences, but to evaluate cognitive performance, and as well as a monitor of continuous brain activity. So let me finish this because I really wanted to listen to you, to your questions and to your ideas, because this is a very open and new field, and just very few conclusions from my side. Clearly, EEG is an appropriate and feasible tool for evaluation functional brain networks during the space flights. There is no doubt about that, and it's going to be used more and more, and it's going to provide much interesting results than the ones I just reported today. And cognitive and motor performance could have a physiological equivalent. We need that, because at the time that the astronauts make failures, that could be a disaster. So we need something that advances how is the brain activity of an astronaut, not to make errors or to have less precision in motor control or whatever. We need that as a predictive value of cognitive and motor performance. And of course, we have found this reduced alpha power and connectivity during flight conditions. This will be taken into account as a potential consequences of a space flight. And of course, we need to develop countermeasures to improve this situation, because if in a potential travel to Mars, which could be at least 9 months, just one way to Mars is could be 9 months. That is a long time period that needs some countermeasures that help the astronaut and the cosmonauts to have a good cognitive and motor performance. And of course, this is just the beginning. We need more data and more research, and it will be coming in following years, I hope, because this is becoming a very interesting and exciting topic. I wanted just to say thank you to the Translational Research Institute for space research in space, because they are supporting some of these studies. And of course, wanted to thank you, this international group that are we are working together in this interesting project. Professor Cheron, that at the beginning provided the data, but now is inspiring us with many ideas, and Professor Patrick Fielder in Elmenau doing all this complex signal analysis, and Professor Funke, who is really the promoter and the heart of all this. So I'm very happy and open to have questions if there is some. There are some.
Thank you very much, Fernando. This is really exciting, and thanks for opening this quite complex and new field in terms of EEG brain recording in space in very complex conditions. So actually, the natural question after your talk, when you were explaining regarding the structural changes, functional changes, what potential interventions and countermeasures could be developed based on your findings and the findings that have been seen in MRI, fMRI, and EEG to mitigate the impact during the space flight, before, after, and actually to mitigate what's going on with the functional brain integrity?
This is really a very good question, because I mean, yeah, we say, okay, there is alpha reduction, there is a change in morphology. What we can do? So many people is doing that right now, doing even animal models and research with the humans as well. There are really good studies on countermeasures like food, countermeasure, chemical countermeasures, physical activity, making psychologically the environment better to reduce the stress and isolation to the astronauts and cosmonauts and so on, and all that helps a lot. But we are coming from the neurophysiology field, and because of that, we are a little bit biased on neurophysiological countermeasures, and that could be perfectly the neuromodulation. We know that the neuromodulation can improve brain activity, can improve alpha brain. I did it to myself recently. I received from my students some series of TACS in the alpha to see if my alpha primes can be improved, and I can write better grants for them. And it's true, and it improves. So that means that probably the neuromodulation could be a really good tool before. I mean, to put the brain in a better conditions for going to the space station, after and during flights, because in these long time missions that are being designed recently, they need something that improves their cognitive functions during the long period of time they have to be on the moon or going to Mars. So yeah, neuromodulation, in our opinion, especially TACS, because it's targeting particular frequency bands, are important for as a countermeasure. But there is no study yet, and there should be some studies in the very near future, I think.
Okay, thank you. We have a question from Frank. Great presentation. At the beginning, you were presenting the data from animals when females actually were more resilient to radiation and building the beta amyloid. So the question from Frank is, if you speculate that the alpha reduction and post-flight recovery would be also different from males, and finally with the application to the astronauts. Yeah. And actually, yeah, the question is, this is happening also to astronauts.
Yeah, very
astronauts and more resilient.
Yeah, very good question as well. Thank you so much for that. Yeah, it seems like the ones that are going to Mars are going to be females because of this animal. Because I mean, they seems to be better prepared for radiation. So probably they are the ones that could be, I mean, better for going to, I mean, based on these animal models. Unfortunately, there is very few female astronauts and cosmonauts. So we couldn't do separate analysis in this alpha to see whether the female and male are different. We need more data. We will do it because this is something really important, and many other researchers from many other fields in this space flight field, they are asking for that, and so we will do it for sure. But there is an important aspect, the alpha reduction. We don't know how is the amyloid in these astronauts. We don't know that. But based in the animal models, if radiation is inducing amyloid, we know from dementia studies, and I have done a lot of electrophysiology in dementia studies. The appearance of the amyloid, it induces at the end a reduction of the alpha power. So it's more or less the same thing we are finding in some other studies where the amyloid is present. So yeah, could be that alpha is associated to that, and females are better prepared for being protected to radiation for some reason I don't really know yet, but there is some hypothesis about that.
Okay, thank you. You are presenting clear data with default mode network and changes in alpha band power and functional connectivity. There is clear impact on astronauts' cognitive abilities, actually. And this is during the flight, upon returning also, and you were mentioning also that the recovery can take very long time, but actually there is an impact during the flight. So there should be any measures taken actually during the flight to counterbalance this effect. What's going on with the cognitive abilities actually of all astronauts?
Mm-hmm. This is a very interesting question as well. This alpha rime is just a neurophysiological parameter, not correlated with cognitive performance yet. But there is a group developing a neuropsychological test, especially for astronauts. You can't use typical neuropsychological or typical neuropsychological tests we use for patients with brain damage on Earth. These guys are special, and they need a more tailored task cognitive task to really evaluate their cognitive performance. So there's a group developing this, and they are very good task already developed. And yes, they are doing these studies as well in the ISS. So they are monitoring cognitive functions, and we could, in the near future, we could correlate our neurophysiological findings with this cognitive performance. As well, we will do a cognitive task during the EEG recording as well to see
to compare actually the behavioral responses with real changes in EEG. I would just switch a little bit actually to the technology. So we were explaining different EEG technology and constraint actually what we have. What would be, from your point of view, the ideal setup in the out of terrestrial environment? What challenges you expect regarding size, weight, power constraint? Actually, you were mentioning already that this is the environment is actually much better regarding artifacts than the standard environment. But regarding like EEG equipment, or maybe we could go to also MEG, maybe OPT, magnetometers somewhere there, or NIRS recording.
Yeah. Well, there could be, in the future, OPMs, these optical pumped magnetometers could be used, but they still need some sort of isolation from electromagnetic noise, and that could induce some problems in the ISS. So, but the technology is advancing, and probably that could be an option. But the only option now is EEG and probably NIRS, but as you said, the complexity of the equipment could be a problem, because the weight of the system itself is a very important issue in the ISS. So that you can't get there an equipment which is very heavy, or is needs a lot of space, or or need a lot of complexity. I mean, they need something really simple, very not very heavy, and very easy to allocate to places or to some place. So it's sometimes it looks like very stupid questions, like how big it is, but it's very important there because everything is really tailored and very well.
Okay.
So this could be problems that the EEG nowadays the EEG technology is very small, very light, and very easy to allocate to any place. So that's why EEG is ideal. NIRS, I mean, if it's possible, that could be an option as well.
So you mean that, like, based on the studies that you were analyzing already, the pre and post EEG was done with different device actually than the real during flight? Because
no, it should be done. I mean, what we are designing now, it should be done exactly with the same EEG system before, in flight, and after. It should be done exactly with the same system to avoid precisely these differences between devices.
But I mean the device that it's right now available in the space flights, there is the device custom made. It's not the device that it's somehow manufactured by anyone.
But that's why we have, there should be a new EEG device. I mean, a more standard EEG device for these studies. Because the one is in the ISS now is a little bit different. Yes.
Perfect. Thank you, Fernando. This was last question. Actually, we are perfectly on time. We're a little bit late with 5 minutes. This was really very interesting. Good luck with all your studies, and hopefully we can have you here again with some news that you will be able to present soon regarding the results from the EEG in the space. And to everyone, I hope you are gonna join us again for next webinars. Actually, on October 26th, we'll host Dr. Zhou from Harvard University, and he will be talking about how we can enhance balance and gait in aging with non-invasive brain stimulation. So a little bit different topic, but again within the neuromodulation field. So please again visit our webpage neuractive.com, where you can register for the upcoming webinars. And thank you again, and have a great day, and see you next time. Thank you very much, Fernando. Thank you again.
Thank you. Bye-bye.
Thank you. Bye.